All right, now we're going to be talking about Chapter 2. That's atoms, ions, and molecules. We'll be learning a bit of chemistry in this chapter. So first we have to talk about matter. Matter has mass and occupies space. It composes the human body. In fact, it composes all material things in the universe. The atom is the smallest particle of matter exhibiting chemical properties of an element. There are three forms of matter. There's solid, liquid, and gas. Going further, atoms can be are composed of three subatomic particles. Neutrons. Neutrons have a mass of 1 atomic mass unit, and they have no charge. Protons also have a mass of 1 atomic mass unit, but they have a positive charge. Electrons are 1/1800th of the mass of a proton or neutron, so they're extremely small. They have a negative charge, and they're located at varying distance from the nucleus in regions called orbitals. Sometimes we also talk about electron shells. Electrons are so small that their mass is considered negligible when computing the atomic mass. The periodic table. So here's how to read an element on the periodic table. It'll start, the big letter in the middle, or letters in the middle, are the chemical symbol of the element. So this is carbon. Chemical symbol is C. You can see the atomic number. That's the number of protons in an atom of the element. And how many protons are in an element is what determines which element it is. Elements are arranged in increasing atomic number within the rows of the periodic table. And then the average atomic mass, which is the sum of protons and neutrons in the element. Here's the periodic table. You can see each element has its own unique chemical symbol. Here is hydrogen, atomic number 1, because it has 1 proton. You go over here, here's helium, atomic number 2, because it has 2 protons. Then we go down to the next row, you'll see 3, 4, 5, 6, 7, 8. So it increases from left to right. The elements outlined here in these colored squares are the elements that we'll primarily be focusing on during this course. So it's not a bad idea to just kind of familiarize yourself with these symbols and which element they're associated with. And you may do a little just review of each element and kind of what it's involved in. For example, sodium is involved in osmosis throughout your cells. So it's also an important electrolyte. Here it is again, the most common elements in the human body, and these are the elements we'll primarily be talking about. Tells you how much of your percentage body weight each element makes up. So as far as diagramming atomic structures go, you're going to put the protons and neutrons in the middle because they're housed in the nucleus of the atom. And then an atom has shells of electrons surrounding the nucleus. So here you have the nucleus of the atom, which contains protons and neutrons. And then circling around it are these electron shells. This is the innermost electron shell. And this is its outermost electron shell, also called the valence shell. Each shell can hold a limited number of electrons. So for the innermost shell, the maximum electrons that can be housed in this shell is 2. Shells closest to the nucleus must be filled first before moving outwards to the next shell. And the next shell usually can house up to 8 electrons. In fact, most of the time in the elements that we'll be talking about the valence shell having 8 electrons in it. Elements tend to lose, gain, or share electrons to obtain a complete outer shell with 8 electrons. This is known as the octet rule. So like I said, these elements that we'll be talking about in this course like to have 8 electrons in their outermost shell. That's the octet rule. Now there are some metals, some transitional metals that do not obey this rule. Just be aware of that. But for all intents and purposes in this course, we'll follow the octet rule. All right, how many valence electrons do the noble gases contain in their valence shell? Well, the noble gases are extremely stable, and that is because they have 8 electrons in their valence shell. They don't tend to interact with other molecules, or other atoms, I should say. An unknown element is found to contain 45 protons and an atomic mass of 64. What is its atomic number? Well, remember, atomic number is how many protons are in the element, so its atomic number would be 45. All right, so covalent bonds. This is where that octet rule really comes into play. Carbon normally just has 4 electrons in its valence shell, so it wants to bond with other atoms in order to add more electrons to its valence shell to satisfy the octet rule. It can be obtained in a number of ways. So I'm going to go kind of into detail on each one of these images here. So here you've got carbon. Note that it has, its inner shell is filled with 2 electrons, and then its outer or valence shell is filled with 8 electrons. And the reason that happened is carbon is sharing or borrowing 1 electron from each hydrogen. So here is carbon's electron and here's hydrogen's electron. Here's carbon's electron, here's hydrogen's electron. And you'll notice that they kind of combine their valence shells right here, a little overlap. And that's how they're sharing these electrons equally between each other. This is an example of single bonds, and this molecule is methane. This is an example of a double bond. The molecule is carbon dioxide, and you'll see carbon again has 2 electrons in its innermost shell, but in its outermost shell, it has 1, 2, 3, 4, 5, 6, 7, 8. So it has completed the octet rule, but what it's doing is instead of just sharing 1 electron with oxygen, it's sharing 2 electrons. So a pair of electrons is shared in a double bond. And this is just another diagram showing the different bonds that can occur in an organic molecule here. And so this is all single bonds. And if you look here, there's 1 pair of electrons being shared between carbon and hydrogen, 1 pair of electrons being shared between carbon and oxygen, 1 pair of electrons being shared between hydrogen and oxygen. So these are all examples of single bonds. If you were to have a molecule with triple bonds, you would see 3 pairs of electrons shared between the atoms. Okay, so chemical compounds. These are stable associations between 2 or more elements that are combined in a fixed ratio. You can classify it as either ionic or molecular. Ions are produced from the loss or gain of electrons. So we have 2 types of ions. We have cations, which are positively charged, and that happens when the atom loses an electron, right? Because electrons have a negative charge. And there's some examples there for you: sodium, potassium, and calcium. And you have anions. These are negatively charged ions, and this happens when an atom gains an electron. Examples given are bicarbonate and chloride. All right, so this is a table of common ions. These 2 tables, this is cations, this is anions. Common ions found throughout the body and what the purpose of those ions are. So sodium, for example, the sodium ion, is the most common extracellular cation, and it, among other things, participates in conducting electrical signals in nerves and in the muscle. It participates in osmosis. And then you can build a sodium gradient. Your cells sometimes build a sodium gradient, which is involved in co-transport of other substances across a membrane. For example, there's a sodium glucose co-transporter in your kidney that's involved in the resorption of glucose from your urine. So highlights what I really want you to focus on: the sodium ion. Know that it's the most common extracellular cation, whereas the potassium ion is the most common intracellular cation. I want you to know that calcium is involved in muscle contraction and the formation of your bone and teeth. It can also be a messenger in hormonal signaling. Hydrogen, important to know that concentration of hydrogen determines the pH of the body fluid that you're studying. Chloride, not only a component of stomach acid in the form of hydrogen chloride, also alters nerve cells' responsiveness to stimulation. Bicarbonate, you should know, buffers pH in blood because it is a base. Phosphate, you should know, is a component of ATP, which is the energy currency of the cells, and it's also a component of nucleic acid, DNA and RNA. It's the most common intracellular anion, and it's a component of phospholipids, which these form membranes we will learn about. Okay, ions in general. Atoms with 1, 2, or 3 electrons in the valence or the outermost shell become cations. For example, calcium has 2 electrons in its outer shell. So let me see if I can draw this for you. Calcium has, here's the nucleus. Here's the inner shell, which has 2 electrons in it, right? Here's the middle shell to calcium, and it's complete. It has 8 electrons in it. Every shell has to be filled before you can go to the next shell. So we know this shell has 2 electrons, and this shell has 8 electrons. Now, we go to the outermost or valence shell of calcium, and it only has 2 electrons, whereas it wants 8 electrons. Well, it's easier for it to donate 2 electrons than it is for it to gain 6 electrons. So calcium donates 2 electrons and develops a charge of +2 because it lost 2 negative charges. Atoms with 5, 6, or 7 electrons become anions. So let's say chlorine. It has 7 electrons in its outer shell. It's so much easier for it to gain 1 more electron and complete its outer shell than it is for it to lose 7 electrons. So it gains 1 electron and develops a charge of -1. Ionic bonds. Cations and anions are bound by electrostatic forces, and they form salts. Let me see if I can show you what I mean. Okay. So the sodium atom in sodium chloride loses 1 outer shell electron and becomes a positive sodium ion. And it loses that outer shell electron to a chlorine atom, which then becomes a chloride ion. Sodium and chlorine ions are held together by ionic bonds in a lattice crystal structure called an ionic compound. So to show you what all of that means, here's sodium. Here's its outer shell. It only has 1 electron in its outer shell, so it wants to donate that electron. Here's chlorine. It has 7 electrons in its outer shell, so it wants to gain that electron. When you go over here, they're bonded, even though you can't see the bond. You notice these are still separate they're bonded by electrostatic forces because sodium is now positive and chloride is now negative. See how the ions are not sharing the electrons. Instead, the bond is formed through electrostatic action between the ions. Whereas if you go back to the covalent bonding, you'll see the electrons are shared. See how the valence shells come together, and those electrons are shared. That doesn't happen in an ionic compound. Okay, molecular and structural formulas. The molecular formula indicates the number and type of atoms. So carbonic acid, for example, is H2CO3. The structural formula not only indicates the number and type of atoms, but it also indicates the arrangement of atoms within the molecule. So you can see here, you could call carbon dioxide CO2, or you could also draw it out to show the double bonds that carbon forms with oxygen, which you can't see in just the regular notation. This structural formulas allow differentiation of isomers, and isomers have the same number and type of elements, but they are arranged differently in three-dimensional space. This is a good example. So glucose versus galactose versus fructose. They all have the same molecular formula, C6H12O6, but their atoms are arranged differently. So here, between glucose and galactose, you'll notice this carbon has a hydrogen above it and a hydroxide below it, whereas this carbon has the hydroxyl above it and the hydrogen below it. Fructose takes an entirely different ring structure shape. It has 5 elements in a ring, whereas galactose and glucose have 6 elements in a ring. And the important thing to know about this is that 3D structure of the molecule really determines its function or how it interacts with other molecules and atoms. So glucose and galactose and fructose all have slightly different tendencies than one another due to this difference in 3D arrangement. All right, covalent bonds are the strongest type of bonds, and this is where atoms share electrons. I showed you that a little bit earlier. Let's see, bring that up. Covalent bond here. So these are examples of covalent bonding. Covalent bonds occur when both atoms need electrons. And then the ones that we'll see commonly formed in the human body will use hydrogen, oxygen, nitrogen, and carbon. Okay, so nonpolar and polar covalent bonds. You can further differentiate covalent bonding, remember, where the atoms are shared between, the electrons are shared between 2 atoms. Well, you can separate covalent bonds into nonpolar versus polar covalent bonds. And the whole concept of this relies on electronegativity. Electronegativity is the relative attraction of each atom for electrons. So it determines how the electrons are shared in a covalent bond, right? A covalent bond just means sharing of electrons. It doesn't necessarily mean equal sharing of electrons. So 2 atoms of the same element that have equal attraction for electrons will form a nonpolar covalent bond, whereas sharing of electrons unequally, which occurs between 2 atoms that have different electronegativities, will result in a polar covalent bond. To understand electronegativity, let's look at the periodic table of elements here. So, increasing electronegativity. As you move from left to right, the elements increase in how much they want electrons. And as you move from bottom to the top, the elements increase in how much they want electrons. So electronegativity increases. This is for two reasons. One, as the elements go from left to right, there are more protons in the nucleus, and that causes an increased attraction of electrons. And as you go from bottom to top, the outer shell, or the valence shell, gets closer to the nucleus. So here we'll show the difference between fluorine and sulfur. Look at the outer shells. Fluorine's outer shell is much closer to its nucleus than sulfur's outer shell. And fluorine actually happens to be the most electronegative element. So we already mentioned nonpolar covalent bonds. This is where electrons are equally distributed between the two identical atoms. Then you can have polar bonds. Electrons are shared unequally between two different atoms. And again, this is due to a difference in electronegativity of the two atoms. Electrons may be pulled more toward one atom than another, and this happens when there's a difference in electronegativity. It has positive and negative poles, and oxygen, nitrogen, and phosphorus have a tendency to pull electrons towards themselves. It's actually really important to remember this because that'll give you a sense of whether a molecule has polarity. So especially oxygen, we see polar molecules with oxygen all the time, but nitrogen as well and phosphorus as well. All right, this diagram should show you a little bit better what I'm talking about. So oxygen is more electronegative than hydrogen. They're sharing electrons, but oxygen is pulling the electrons closer to itself, and that makes it slightly negative compared to the hydrogen, which is now slightly positive. And that's what these symbols mean. Delta with a negative after it, that means partial charge of negative. Delta with a positive after it means partial charge of positive. The relative differences of these charges is what influences polar molecules to bind the way they do. And we'll learn about that here in a little bit when we talk about hydrogen bonding. So just keep this diagram kind of in the back of your mind for now. Okay, and then as we stated before, in nonpolar molecules, there is little difference in electronegativity between the atoms, so the electrons are shared more equally than in polar bonds. And you will likely be asked on the exam to identify a polar covalent bond versus a nonpolar covalent bond. So I would definitely familiarize yourself with this concept and come to me with any questions you have. I'm happy to answer them. Here's an example of polar molecules, and you'll see here the oxygen with its partial negative charges, the hydrogens with their partial positive charges. The glucose molecule, look at these oxygens hanging out, and they've got a partial negative charge. A covalent bond occurs when two atoms bond together, one electron transfers from one atom to another, forming two ions. Both bonding electrons come from the same atom, or an electron pair is shared between two atoms, creating the bond. D. Covalent bonding is all about electron sharing, whether it be equally or unequally. Remember that ionic bonds are formed by electrostatic forces and not by sharing of electrons. Okay, of the following, which is not a type of chemical bond? We have ionic, that's definitely a chemical bond. We have covalent, that's a chemical bond. We have polar, that's a chemical bond. So magnetic is not a chemical bond. Magnetic forces do not have anything to do with sharing electrons. Which of the following molecules exhibits ionic bonding? Sodium chloride, carbon dioxide, C6H12O6, remember this could be one of many isomers, or H2O. Ionic bonding, that's when two ions come together through electrostatic forces and form a lattice, and that happens with salts. So sodium chloride is a salt, and it is the correct answer to this question. All right, amphipathic molecules. These are large molecules that have both a polar and a nonpolar region. One of the most classic examples is phospholipids. So you can see phospholipids have sort of this head region here that is electrically charged with the nitrogen and the oxygen carrying charges. And it has a tail region that has long chains of carbon and hydrogen. And these are covalent bonds. There's no polarity in these, so these are nonpolar bonds. And this shows you a little bit more in detail. So what tends to happen with phospholipids when you put them into a liquid? The head regions that are tend to orient themselves toward the water, while the tail regions tend to orient themselves away from water, right? Because the heads are hydrophilic or water-loving, they're polar, and they want to interact with a polar molecule such as water. Whereas the tails, they are nonpolar, so they want to interact with water as little as possible. How they achieve this in nature is they form a natural bilayer. So what happens is the heads orient themselves out towards the water, and the tails orient themselves inwards, and you get two layers of phospholipids arranged like this, and it creates membranes in living systems. All right, next we're going to talk about intermolecular attractions. These are weak chemical attractions between molecules, and it's important for the shape of complex molecules. Note that intermolecular attractions don't have to do with sharing electrons. So first we'll start with the hydrogen bond. This forms between polar molecules. It's an attraction between the partially positive hydrogen atom and a partially negative atom. They're individually weak and collectively strong, and that influences how water molecules behave in particular. So here's a schematic of hydrogen bonding, water bonding with glucose. Now remember that the oxygen is more electronegative, so it tends to pull the electrons towards it. That means the oxygen atom has a region of partial negativity over it. And this interacts with this hydrogen atom that has a partial positivity over it because this oxygen is pulling its electrons away from it. So this hydrogen is partially positive and interacts with the partial negativity of the water molecule. And you can see just one hydrogen bond is not that significant, but you can have multiple hydrogen bonds at the same time. Van der Waals forces, this is the next intermolecular attraction we'll talk about. These occur in nonpolar molecules. So what happens is electrons orbiting the nucleus briefly become unevenly distributed. And this unequal distribution can induce another area of unequal distribution in an adjacent atom of another nonpolar molecule. Individually, these are also weak. So let me show you what this looks like. Here's atom number 1, or A. This is its nucleus, and this is the cloud of electrons orbiting it. At any given time, these electrons could be on one side of the atom more than the other, or they could be evenly distributed. It's really just random chance. So randomly, this atom becomes more negative here on this side. You can tell there's more electrons on this side, so there's a partial negativity over here. On the other hand, there's a partial positivity on this side because there are fewer electrons. What can happen is atom A can go on to induce the same situation in atom B. You'll see that atom B starts up here with pretty equal distribution of its electrons. Oh, that's my cat French fry. Anyway, atom B has an even distribution of electrons at this moment in time. But atom A, due to its partial positivity, draws those electrons to one side of atom B, inducing a small dipole in atom B. Van der Waals forces are about 1% the strength of a covalent bond, but there are millions and millions of these interactions, so they add up and can be quite strong. The classic example I was always given was a gecko. The reason that it's able to stick to the ceiling is because of van der Waals forces between its toes and the ceiling itself. Hydrophobic interactions, this happens when nonpolar molecules are placed in a polar substance. Nonpolar molecules arrange themselves together to reduce interactions with polar water. And this can happen on a larger scale. For example, if you had a large molecule, and when it happens in a large molecule, it's termed intramolecular attractions. This is an example of hydrophobic interactions. So you've poured oil on top of a cup of water, and the oil tends to float. And why is that? Well, it wants to reduce its interaction with water, and it does that by floating to the top, so that it only has to interact with water in this one plane. If the oil stayed down here in the middle, it would be interacting with the water all around it, and that would increase the interface between the nonpolar molecules and the polar water. So to reduce that, oil floats to the top. All right, molecular structure of water. Water composes 2/3 of the human body by weight, so it's a pretty important molecule to know about. We've talked about how it's one oxygen atom bonded to two hydrogen atoms, and that oxygen atom has partial negative charges, right? One partial negative charge from the first hydrogen atom that it's pulling electrons away from, and another partial negative charge from the second hydrogen atom that it's pulling electrons away from. For that reason, water can form 4 hydrogen bonds with adjacent molecules. And this is really what gives water all of its unique properties. Let me see if I can find the image. Here. Here's an example of water being able to form 4 hydrogen bonds. So this hydrogen can form a bond, this hydrogen can form a bond, and the oxygen can form 2 bonds, 2 hydrogen bonds. Functions of liquid water are transports, so substances dissolve in water and move more easily throughout the body. It lubricates, it decreases friction between body structures. It cushions, so it can absorb sudden force of body movements. And it excretes wastes, so unwanted substances can dissolve in water and be eliminated. Water is the universal solvent of the body. Solutes are defined as substances that dissolve in water, and the chemical properties of a substance determine whether it will dissolve or not. We already saw that with oil in water, oil being a nonpolar substance, therefore it does not want to dissolve into a polar substance like water. Substances that dissolve in water, polar molecules and ions. They're both hydrophilic, which means water-loving, and the water surrounds the substances and forms a hydration shell. So you can see right here is glucose, and it's got those hydrogen bonds from water surrounding it, and you see how water kind of creates a shell around the molecule, and this is a hydration shell. You also see hydration shells with ions. Note that these are not hydrogen bonds. This is just electrostatic attraction. Some substances dissolve and dissociate or separate entirely. And you see that with sodium and chloride here. It dissociates into the sodium and the chloride ions. These are known as electrolytes, and they can actually conduct current through the body. Substances that do not dissolve in water, these would be nonpolar molecules. So hydrophobic, meaning water-fearing. Hydrophobic exclusion, this happens when cohesive water molecules force out nonpolar molecules. It's kind of like the other half of hydrophobic interactions, right? Hydrophobic interactions is when the nonpolar substance wants to interact with itself and decrease its interaction with water. Well, water doesn't really like nonpolar substances either, so they do their own thing where they exhibit hydrophobic exclusion, where the cohesive molecules, so the ability of water to attract itself, forces out those nonpolar molecules, so they really have no place to go. This is important to note because hydrophobic substances generally require proteins to be transported within the blood. So they can't dissolve in the blood, so they hook onto a transport protein or a carrier protein to be transported within the blood. Now, many substances partially dissolve in water, and we talked about this a little bit earlier, the amphipathic molecules that have polar and nonpolar regions. So the polar portion of the molecule will dissolve in the water, but the nonpolar portion is repelled. We talked about this. Phospholipid molecules, polar heads have contact with water and the nonpolar tails group together. This results in bilayers or the formation of cell membranes. There's another arrangement that phospholipids can take, and this is called the micelle. And if you look closely, the heads are all pointing outward and the tails are inward. You'll see micelles when it comes to digestion of fats. What happens is a fat molecule will get trapped inside here where it gets to interact with all the other nonpolar substances just like it, while the heads of the phospholipids interact with water and remain, keep everything dissolved. So what you definitely need to know are hydrophilic substances dissolve in water. Hydrophobic substances do not dissolve in water, and amphipathic substances partially dissolve in water. This is just showing all the different roles that water takes in your body. Like cushioning, there's cerebral spinal fluid that helps cushion your brain. Water helps regulate your body temperature. It's a universal solvent. It helps, it lubricates. It's involved in acid-base reactions. All right, now we're going to talk about water mixtures. So a suspension is a mixture composed of large particles. A colloid is a mixture composed of smaller particles than a suspension that remain in solution when not in motion. So a suspension will fall out of solution when it's not in motion. You have to swirl it to sort of dissolve all of those particles. A colloid has smaller solutes, but they don't settle. Whereas a solution is a homogeneous mixture of dissolved solute and water. So that's like we saw with the glucose being dissolved in water and water forming a hydration shell around it. An emulsion is composed of water and a nonpolar substance like vegetable oil and water. You would find that in something like a salad dressing. A salad dressing is an example of an emulsion. It will not mix unless you shake it. All right, acids and bases. This is a lot to take in, but we'll get through it hopefully pretty straightforward. An acid dissociates in water to produce hydrogen ions and a correlating anion. It increases the concentration of free hydrogen ions that are available to interact with other molecules. On the pH scale, a pH of 1 to 6 is considered acidic. More dissociation of hydrogen is seen with stronger acids. That makes sense, right? So the stronger an acid is, the more likely it is to give up that hydrogen ion. And here's the equation for it. Substance A, which is an acid in water, dissociates to a hydrogen ion and an anion. Bases, instead of donating hydrogen ions, accept hydrogen ions when added to a solution. This decreases the concentration of free hydrogen ions there are to interact with other molecules. On a pH scale, a pH of 8 to 14 are basic or alkaline, and there's more absorption of hydrogen ions with stronger bases. So ammonia and bleach would be an example of strong bases. And this is showing substance B plus a hydrogen ion go to this molecule where B and H are attached. Okay, so what is pH? pH is a measure of the concentration of hydrogen ions in the solution. It's measured on a scale between 0 and 14, with 0 being towards the acidic end and 14 being towards the basic end. pH and hydrogen concentration are inversely related. So that means as pH increases, for example, as it goes from 8 to 9, hydrogen ions decrease. So there are fewer hydrogen ions. That makes sense. A solution of pH 9 is basic, so it doesn't have a lot of hydrogen ions. As far as the math goes, it's the inverse of the log for a given hydrogen concentration. So inverse of the log, so inverse meaning that as hydrogen concentration increases, pH decreases. And the log means that we're working on a scale of base 10. And to simplify that, you can say that every increment on the pH scale increases by a factor of 10. So let's say we start at neutral, which is pH 7, and you go to a pH of 6. pH of 6 is 10 times more acidic than a pH of 7. A pH of 5 would be 10 times 10 or 100 times more acidic than pH 7, and a pH of 4 would be 1,000 times more acidic than a pH of 7. This works in the opposite direction as well. So a pH of 10 would be 7 to 8, 10, 10 times 10 times 10, 1,000 times more basic than neutral. This is just a pH scale giving examples of some substances. What I want you to know for sure is human blood is almost neutral. It's there at 7.4. Pure water is at 7, and that's true neutral. And then just familiarize yourself. For example, hydrochloric acid has a pH of 1, so it's extremely acidic. And its action is in the stomach, which makes sense. You want an acidic environment to help digest your food. What is the most likely pH of a solution containing many hydroxide ions, so bases, and few hydrogen ions? Okay, we don't have to do any math to figure this out. pH of 2 is acidic. A pH of 6 is acidic, so there's going to be more hydrogen ions than hydroxide ions. In a pH of 7, it's true neutral, so exactly however many hydroxide ions there are, that's how many hydrogen ions there are. So the answer would be D. This is the only one of the answers that is a basic number. The concentration of hydrogen ions in a neutral solution is, this is neutral. So we're going to look, we're looking for base 10 log, right? So we're going to look at these three here because these are bases of 10. And remember, it's the inverse log. So it would be B, 10 to the negative 7, because your pH is 7. But remember, it's the inverse of the log, so it's 10 to the negative 7. All right, next we're going to start talking about biological macromolecules. And let's just go over some of the general characteristics. First, we need to know what an organic and inorganic molecules are. So an organic molecule is a molecule that contains carbon, more specifically, a molecule that contains carbon bonded to hydrogen. Most are a component of living organisms, and among the component, a subset are biological macromolecules. Inorganic molecules would be all other molecules. So water is an inorganic molecule because it does not contain carbon bonded to hydrogen. Carbon dioxide, even though it contains carbon, is not organic because it does not have carbon bonded to a hydrogen. The four classes of organic biomolecules in living systems are lipids. These are your fats. Carbohydrates, these are things like sugars, starches. Nucleic acids, this is your DNA and your RNA. And proteins, and remember, proteins are made up of building blocks called amino acids. All right, now for just a little bit of chemistry. First, we're going to talk about a dehydration synthesis. This is also called condensation. This occurs during the synthesis or building up of biomolecules. So you'll see one subunit loses a hydrogen and another subunit loses an OH. And a new covalent bond is formed and water is produced. So this is the macromolecule that you're building. These are the subunits you're building it with, and the subunits lose water. Put off water, and you're left with these two combined. So let's say this was two glucose molecules, and you're trying to make glycogen. So these glucose molecules come together, give off water, and what's left behind is a part of glycogen that you can continue building on. Hydrolysis is just the opposite. It occurs during the breakdown of biomolecules. And as the name implies, it involves the lysing or splitting of water and adding it back to those subunit molecules. All right, so the first type of biomolecule that we're going to talk about, macromolecule that we're going to talk about, are the lipids. This is made up of carbon and hydrogen with very little oxygen. It's a diverse group of fatty, non-polar, hydrophobic molecules. The main functions of lipids are stored energy, the cellular membrane, and hormones. And there's four primary classes we're going to talk about. The first one is triglycerides, so that's going to be the primary storage form of lipids. 2, phospholipids, and we've already touched on these, the primary constituent of cell membranes. 3, steroids. In the human body, these act as hormones or signaling molecules. And 4, the eicosanoids. These are also a local signaling molecule, so they work in very short range ways. All right, so first up is triglycerides. This is the most common form of lipid. This is the long-term energy storage that we see in adipose tissue. It's composed of a glycerol molecule and 3 fatty acid tails. The fatty acids vary in length, and they vary in number of double bonds. So if you have a saturated fatty acid, it's going to have all single bonds, no double bonds whatsoever. If you have an unsaturated fatty acid, on the other hand, it's going to have it's going to have 1 double bond. And if you have a polyunsaturated fatty acid, it has 2 or more double bonds. Adipose tissue, we'll talk about lipogenesis, which is the formation of triglycerides when conditions of excess nutrients exist. So you have too many nutrients and you need to store them for later so you don't lose the energy in those nutrients. Lipogenesis, so the formation of triglycerides, is what will happen. On the other hand, let's say that you are starving and you need nutrients. Well, you need to break down those triglycerides, and that's known as lipolysis. All right, on top, this is showing a glycerol molecule here and 3 fatty acids here. The reaction moves in this way, and we build a triglyceride. So what happens is we bond this glycerol molecule to these 3 fatty acids. In doing so, water is given off. So is this a hydrolysis reaction or is this a dehydration synthesis? Well, it's a dehydration synthesis, and you know this because water is given off, but also because you're synthesizing something. You are making a triglyceride. This is known as lipogenesis. The opposite direction is lipolysis, where you break down the triglyceride into its constituents, and that occurs through hydrolysis, or the splitting of water. The other classes of lipids you need to be familiar with are the phospholipids. We already talked about these a bit. These are the amphipathic molecules that form cell membranes. The phospholipid structure is similar to a triglyceride, except it's a phosphate group that's linked to the glycerol head with 2 fatty acid tails. The eicosanoids, these are 20-carbon fatty acids with a cyclic hydrocarbon group. And that's what that looks like here, the eicosanoids. So 20-carbon fatty acid, these act as local signaling molecules. So the thromboxanes, the leukotrienes, the prostacyclins, and the prostaglandins. A lot of this is involved in the inflammatory response. And then steroids. These consist of 4 carbon rings attached to each other, and these include cholesterol, steroid hormones, and bile salts. And this is what they all look like. We already went over the triglycerides here, the glycerol head and the 3 fatty acid tails. Here's what a phospholipid looks like, and you may recall the 3D structure earlier in this lecture that we saw, but the polar head and the nonpolar tails. Polar head, nonpolar tails. Steroids, here are the rings of the steroids and the eicosanoids. Okay, carbohydrates. Carbohydrates in the human body function as the main energy source. In a carbohydrate, hydrogen and hydroxyl are usually attached to every carbon. So that's where the hydrate part of carbohydrate comes in. It's essentially a water molecule attached to every carbon. The chemical formula for carbohydrates is CH2O, and you multiply that by N, which is the number of carbon atoms in the molecule. When we talk about carbohydrates, we can talk about monosaccharides, which are the simplest sugar monomers. We could talk about disaccharides, which are formed from two monosaccharides. Or we could talk about polysaccharides, which are formed from many monosaccharides. Over here you have some monosaccharides. And down here you have disaccharides. So this is a single sugar molecule, and this is two sugar molecules bonded together. The most important carbohydrate that we'll talk about is glucose. It's a monosaccharide. It has 6 carbons in it, and it forms this ring here. It is the primary nutrient supplying energy to our cells. Glycogen is an important storage molecule. It is a polysaccharide made out of repeating glycogen monomers. It's large and branching, and it's stored in the liver and skeletal muscle. So here's a glucose molecule. When it goes to form glycogen, that's called glycogenesis. And that happens in periods when what? You are nutrient rich, so you have an excess of glucose and you want to store it. So you go through glycogenesis. Then, during periods where you are energy poor and you need that glucose back, you can break down glycogen, called glycogenolysis. Nucleic acids. This will be DNA and RNA. So these store and transfer genetic information. DNA is the storage form, and RNA is the transferring form of nucleic acid. These are made of nucleotides, and a nucleotide is a sugar, a phosphate, and a nitrogenous base. The two types of nucleic acids are deoxyribonucleic acid and ribonucleic acid, and they are so named because in DNA, the sugar that's present is deoxyribose, whereas in RNA, the sugar that is present is ribose. Nucleic acids are made up of those of repeating nucleotide monomers that we just saw, the sugar, the phosphate, and the nitrogenous base. The sugar is always a 5-carbon pentose. This gets down in the weeds a little bit, but the phosphate group is attached at carbon 5 of sugar. So when you label sugars, you have this oxygen here, and the first carbon is out to the right of that. The second carbon, the third carbon, the fourth carbon, and then up here is the 5th carbon. These points represent carbon atoms. Even though they're not drawn, even though the C is not drawn there, these points represent carbon atom. The phosphate group is attached to carbon 5, so that makes it stick out this way, whereas the nitrogenous base is attached to carbon 1. The nitrogenous base has many nitrogens in it, so this is a purine, and it has 4 nitrogens. The pyrimidines are a single ring structure. All right, and these are shown here. So starting with the pyrimidines, you have cytosine. This is in both DNA and RNA. You have thymine, which is only in DNA, and you have uracil, which is only in RNA. Those are the pyrimidines. These are the different types of nitrogenous bases. Down here are the double ring structures, the purines, and there's only 2 of those. Adenine, both in DNA and RNA, and guanine, both in DNA and RNA. And see how we notate these with these symbols here. We'll talk a little bit more about how DNA bonds, and these symbols will come in handy for reading the diagrams. You don't have to memorize these structures, but I do want you to be able to tell a single ring structure from a double ring structure and tell me, is it a pyrimidine and a purine? I also want you to be able to name all of the nitrogenous bases, which other bases they bond with, and tell me the difference between uracil and thymine as far as which one's in RNA and which one is in DNA. All right, so DNA. This is the basis of your genetic code. It is a double-stranded nucleic acid, and it is located in the chromosomes in the nucleus of a cell, and the mitochondria of a cell also have their own DNA. Like we said, it's composed of a deoxyribose sugar, a phosphate, and one of the 4 nitrogenous bases that DNA uses. So that would be either adenine, guanine, cytosine, or thymine. And the double strands are held together by hydrogen bonds. So let me show you here. You can see the strands of the ladder are composed of nucleotides. So, right, that's a phosphate, a sugar, and a nitrogenous base. So here's one nucleotide, 2, 3. They're bound together through, I believe, they're phosphodiester bonds. Over here is a nucleotide, nucleotide, nucleotide. Now look at how they're pointed in different directions. This one is moving this way, and this one is moving this way. And then in the middle, their nitrogenous bases line up, and they form hydrogen bonds between them. Adenine and thymine have 2 hydrogen bonds. Cytosine and guanine have 3 hydrogen bonds between them. Which one do you think has a stronger adherence to one another, cytosine and guanine or adenine and thymine? Well, cytosine and guanine have 3 hydrogen bonds, so it's a little bit stronger than the bonds between adenine and thymine, just because there's an extra hydrogen bond. RNA, this is the single-stranded nucleic acid located in the nucleus and in the cytoplasm of the cell, right? Because it has to go out there and take instructions on how to make protein from that gene. It's composed of a ribose sugar, a phosphate, and one of the 4 nitrogenous bases that RNA uses. So adenine, guanine, cytosine, and in RNA, there's uracil. This table just shows the major differences between RNA and DNA. I do expect you to know this. One more nucleic acid we'll talk about is adenine triphosphate. This is an energy molecule. This is the currency that your cells use when they need to either use energy or store energy. What it is essentially a nucleotide, but instead of just having 1 phosphate group hanging off of it, there's 3. And these bonds, these extra bonds, are high energy. So what you can do is you can cut this bond and it releases energy. Or if you need to store energy, you can throw the phosphate back on there and you've stored energy in the form of a chemical bond. All right, proteins. These are the most abundant macromolecules in the body. General protein structure is composed of 1 or more strands of amino acid monomers. So that's amino acids attached to one another. There are 20 different amino acids. Each has an amine and a carboxyl functional group. So down here, this is an amino acid. This is the central carbon. Coming off of it is a carboxylic acid and an amine group. That's why they're called amino acids. Down here is a variable group. It can be one of 20 different structures. We'll look on the chart next. What you need to know about this is that protein structure depends on the sequence of amino acids, and the amino acids essentially fold in different ways depending on which R group is there, which the R groups can kind of be attracted to each other, right, through hydrogen bonding. So depending on what R group comes next depends on how the sequence of amino acids is going to respond to it. Amino acids are covalently linked by peptide bonds. There is an N-terminal end, that's this end of the amino acid, and there's a C-terminal end, that's this end of the amino acid. Okay, so here we have one amino acid, and we're going to link it to another amino acid. The C-terminal from one amino acid connects to the N-terminal of the next amino acid through a dehydration synthesis reaction. Remember, this is dehydration synthesis because we are synthesizing a new molecule here. And down here you can see these are the peptide bonds highlighted in purple. These are the R groups hanging off of each amino acid residue that's left in the protein. These R groups may attract one another or repel one another, and it will kink this chain of amino acids and give it a structure. We'll talk about that in a minute. Here are the amino acids, and what I want you to take away from this, I don't want you to worry about memorizing the amino acids. I do want you to realize the structure of one, though. So here's that central carbon. There's the C-terminal end where the carboxylic acid is. There's the N-terminal end where the amine group is. And then down here is that variable R group. So on this amino acid, valine, you can see that there are 3 carbons coming off of it, and they each have hydrogens attached to them. This would be a non-polar R group. It's not likely to want to attract water or other polar substances. It's going to want to interact with other non-polar R groups. Whereas if you go down here, we have aspartate. Again, the central carbon, the C-terminal end, the N-terminal end, and here's the R group. Here we have a charge. In fact, this is a carboxyl group hanging off. So what we know about carboxyl groups so far is that they are polar. They want to interact with other polar molecules. So let's say that we had an amino acid, let's say that we had a little peptide, right? Just made out of valine and aspartate. Well, are those R groups going to want to interact with each other? No, they're going to repel each other. And that will kink that little peptide. These are the various protein functions in the body. It's good to be familiar with the different roles that proteins play. So they can be an enzyme and catalyze or speed up a chemical reaction. The proteins can be fibrous and provide support for the intracellular structures of cells. Actin and myosin proteins are involved in skeletal muscle cells and help with body movement. Proteins can also help with blood transport. For example, your red blood cells are packed full of hemoglobin, and that helps transport oxygen through your blood. Proteins are also part of membrane transport. They can, carrier proteins move polar molecules such as glucose across the plasma membrane. And they're also involved in immunity. Antibodies, specifically, bind pathogens and help fight off disease. What I want you to know about this is that protein function, like what its job is entirely dependent on its structure. Proteins can take many different forms, but what they do totally depends on what their form is. And we call this form their conformation. It's a three-dimensional shape of the protein. It's very important for protein function. It determines protein function. And then the conformation itself, the shape it takes, totally depends upon those intramolecular attractions between amino acids. So a little bit more about intramolecular interactions. There's the hydrophobic interactions with nonpolar amino acids, and they want to be farther from water. Then there's hydrogen bonds between polar R groups, between the amine and the carboxylic acid groups. There's ionic bonds that can form between the negative and positive R groups. And interestingly, disulfide bonds form between cysteine amino acids. So if you had a sequence of amino acids that had several cysteines in it, the likelihood that two of those are going to form a disulfide bond is very high. And the reason it does that is because it has a sulfur on it here. And this sulfur likes to bond with other sulfurs, and that stabilizes the growing protein. All right, protein conformation, we talk about it in terms of four structures. We talk about the primary structure of protein, we talk about the secondary structure, the tertiary structure, and finally, the quaternary structure. So the primary structure of protein is simply the sequence of amino acids. It's which amino acid comes next. Secondary structure, on the other hand, is the spatial arrangement or shape of that sequence of amino acids. So you can see here, these R groups coming off, and then some of the central carbons are hydrogen bonding with other molecules. And this gives the shape of an alpha helix when the hydrogen bonding occurs in this formation. Whereas hydrogen bonds could also cause the amino acid sequence to form a beta pleated sheet, which is kind of this accordion-looking little structure. The alpha helix gives elasticity to fibrous proteins like skin and hair, whereas the beta sheet it gives flexibility to globular proteins, so like proteins that are sort of spherical in shape, roughly spherical, like enzymes. Now imagine that you have an alpha helix and a beta pleated sheet, maybe another alpha helix. Those structures are going to want to interact with each other. So this is tertiary structure. This is when the alpha helix and the beta pleated sheet start to bend even more and create a unique three-dimensional structure. Globular proteins fold into a compact shape. Fibrous proteins are extended linear molecules. So here's the globular protein, and see how it's kind of folded in to make sort of roughly spherical shape. And then here's the fibrous protein, and it's a long strand. An example of a fibrous protein might be keratin. Okay, finally, there's quaternary structure. Now, many proteins stop at tertiary structure, but some proteins go on to form a quaternary structure. And what that is when those proteins with their special tertiary shape want to interact with each other and form kind of like a megaprotein here. You'll see there's 4 different protein subunits that have fit together to form this quaternary structure. You could also do the same with fibrous proteins, where you've got multiple subunits of fibrous proteins intertwining to create a stronger structure. When a protein is functional and working the way it is supposed to, it's said to be in its native state. Denaturization causes conformational change to a protein and disrupts protein activity, and it's usually irreversible. So remember, protein function relies on protein structure. So if you disrupt the structure of the protein, you erase its ability to perform its function. The common causes of this are increased temperature, increased acidity, some alcohols, and some disinfectants. So imagine you're cooking an egg on the skillet, right? And the egg white turns white. What you're doing is you're actually denaturing the albumin that's in the egg white. Which is not a category of protein? A, enzyme, B, triglyceride, C, antibody, or D, cotransporter. Well, we learned A, enzymes are proteins used as catalysts, or in other words, they speed up reactions. We learned that antibodies are also proteins that help in fighting off infection. And so now we're down to B or D, and hopefully you remember that B is a type of lipid and not a type of protein. The cotransporter is an example of a membrane-bound protein that helps transport substances into or out of the cell.